Hello students. So many of the students have requested me to just revise or give the introduction of downstream processing in the fermentation technology. But those have joined very late. I will give a basic introduction to the fermentation technology or the fermentation process so that you will be able to know what exactly is the downstream processing. This is the complete diagrammatic presentation of fermentation process. You can take this diagram and you can go the Steinberg and Whittaker principles of fermentation technology to have more elaborative text on this topic. So basically in fermentation we have upstream processing, we have fermentation, and we have downstream processing. There are three basic aspects. In upstream processing you can see this basic part is in upstream processing where we take the stock culture. It may involve the procurement of the culture, purchasing the culture, the right culture, or it may involve the isolation of microorganism, then screening, primary screening, secondary screening, and the revival of the stock culture if you have purchased the stock culture or you have preserved the culture. So everything that is in the beginning, we need a culture, and then we will revive the culture. Then we prepare the inoculum. For inoculum preparation we need some raw material for media formulation, media sterilization. So all these aspects, inoculum development. So right from the beginning, when we have discussed in the classroom that first step is the isolation of microorganisms, that is a different aspect. Then preservation of the cultures, that is a different thing. And then screening of the microorganisms, the primary and secondary screening, and we perfectly select a desired strain that will be carried to the fermentation for the production of the desired product in the desired yield. Then the central point is the fermentation, the fermenter. And after the fermentation, when the fermentation is over, we have the culture fluid or harvested broth. So first and the important thing in the first after the harvesting the broth is the separation of cells. So the cell separation method is itself a big and interesting topic. We can use the different protocols, different methodologies, different techniques. For example, you can use this form separation method, you can use the precipitation method, you can centrifugation use, you can different types of filtration. It may be the batch filtration, it may be the continuous filtration, but whatever technique you will use, basically we will separate the cells from the broth. So we'll get the two components, one will be the cells and other will be the cell-free supernatant. So if the product is extracellular, then definitely product will be in the broth. So you will preferably we will take the cell-free supernatant. But if the product is intracellular, then definitely we will keep the cells, and we have to disrupt the cells. Again, the cell disintegration, cell disruption is again an important aspect. We can use the different biological available methods, chemical methods, physical methods for the disruption of cells and separation of the product. So whether it is intracellular or extracellular, the end point will be that we have a product into the buffer, in suitable buffer. So after the getting the product into the buffer, we will go for the purification and then packaging, and simultaneously the effluent will be treated according to the norms. So this point up to the fermentation is a different thing, and the rest, the parts or the techniques or the processes which are used after the completion of the fermentation, after the harvesting the broth, will be covered into the downstream processing. So downstream processing particularly leads to the product purification, product extraction, product purification, and then packaging, crystallization. Everything comes into the downstream processing. So we can switch to the next slide. This fermentation broth, removal of solids, cell disruption. If the product is intracellular, then we will disrupt the cells. Then primary isolation of the product, then purification and concentration, then final product. So this is the basically downstream processing. So the removal of solid will have different methods, techniques. Cell disruption will have different techniques. Primary isolation will have different techniques. Purification and concentration are different techniques. And the basic steps, these are the basic steps. We need to elaborate further, and we will take these steps one by one in the next classes. So this is for the introduction and for the students who have joined late. Thank you very much.